All right, our final section is still on that third learning target that students will be able to explain using evidence how meiosis contributes to genetic variation. So this final PowerPoint will actually take us through the steps of meiosis, kind of like we did when we talked about mitosis. So in meiosis, the cells are going to go through two divisions, and this is a very important distinction. They're still going to go through that PMAT, prophase, metaphase, anaphase, telophase, but they're going to go through it twice. So we kind of break it down into what we call meiosis 1 and meiosis 2. The big thing about meiosis is that it reduces the chromosome number in half. So we're going to go from diploid to haploid. Whereas in mitosis, we just had started with diploid cells, we ended with diploid cells, 2 of every kind of chromosome in those cells. And meiosis also creates genetic diversity, which is very important by mixing the chromosomes and the genes. And we'll show you how that happens. So again, a thing to keep in mind about meiosis is that our goal is to produce genetically unique or different cells, not the same ones like we did for mitosis. We're trying to create haploid cells, the gametes, your egg and sperm cells. This takes place only at certain times in an organism's life cycle, so it doesn't continuously happen like mitosis does for growth and repair, and it is involved in sexual reproduction. So when we look at the overall stages of meiosis, we're going to be starting out with diploid cells where we have 2 of every kind. So here's 2 and here's 2. Again, remember we would have 46 chromosomes in our body cells, 2 of every kind. So 23 pairs. And in meiosis 1, that's actually called the reduction division because we go from diploid to haploid. Now we only have 1 of every kind of chromosome instead of 2. So it's already after meiosis 1 that we have reduced the chromosome number from diploid to haploid. They're still replicated chromosomes, but we only have 1 of each kind. And then during meiosis 2, we call that equational division because we go from haploid and we still are haploid. All we did is we pulled apart those sister chromatids, but there's still only 1 of every kind of chromosome in these cells. It's just that now they are unreplicated versus the replicated ones over here. So for meiosis 1, this is going to occur after the DNA has been replicated during the S phase of interphase. So we're still going to have interphase. We're still going to have gap 1, synthesis, and gap 2. So all those things like growth and then the DNA being replicated, that has to occur before we can have meiosis occurring. The big thing I want you to know about meiosis 1 is it divides the homologous chromosomes. Okay, so meiosis 1 is all about the homologous chromosomes and dividing those. So first of all, in prophase 1, the homologous chromosomes pair up. So they get really close to each other. That didn't happen in mitosis when we did that process. But in here, you can see we have, here's a pair of homologous chromosomes, and they're just really close to each other. Same with this one. And when they do that, they can undergo what's called crossing over, where they actually can exchange some genetic material. Your chromosomes are still going to condense, your spindle fibers are still going to form, and your nuclear membrane is still going to break down along with the nucleolus. So those similar things that happened in mitosis are still occurring here in prophase 1 of meiosis. But the big event is crossing over, and we're going to show you what that looks like here in this next slide. So crossing over means that part of the homologous chromosomes actually exchange genetic material. Remember how I told you homologous chromosomes are the same but can have different forms. Here's what I mean by that. Let's say both of these are your number 1 chromosome. Here's number 1, here's the number 1 chromosome. Notice that they have the same genes, A, B, C, D, E, and F, okay? But this chromosome, homologous chromosome, has all dominant genes. They have all the capital letters. And this homologous chromosome has all the recessive genes for those A through F with the lowercase letters. So what can happen during prophase 1 is when those homologous chromosomes pair up, they can actually cross over like this, and they can actually trade tips of their chromosomes. So now look at this chromatid still stayed the same, but this one now has a new combination because it traded the tips, that gene, with the other homologous chromosome, and now this one has a new combination as well. This one stayed the same over here. So if you look at all these, all 4 of these chromatids are different. So crossing over results in new combinations, or what we call genetic variation. So this is one of the main ways that we get genetic variation, is through crossing over. If you have any brothers or sisters, you might look a lot like your brothers or sisters, or you can look quite differently. If you have the same parents, same mom and dad, some people say, well, you know, they're tall and they've got blonde hair and the other one is short with dark hair. That can still happen in the same family. It just depends on how many of these crossing over events occurred, because that can give you whole new combinations of genes that your siblings didn't have, because they had different crossing over events when the egg and sperm were going to be forming for them. Metaphase 1, the homologous pairs line up in the middle just like they normally do, just like they did for mitosis, except in mitosis you didn't have the pairs line up. You only had just one individual one spread, so you would have had 46 chromosomes across, replicated chromosomes across the middle. Now we have the homologous pairs lining up in the middle like that. So they're still right by each other. And we can see here, there's some crossing over that occurred, there's some crossing over that occurred in those. Now, when these chromosomes line up in pairs across the middle of the cell, they line up randomly, okay? And so because they line up randomly, how they're going to get pulled apart is totally different. You might get different combinations going one direction and then you get going the other direction of those chromosome pairs. And we call this independent assortment. And again, this is the second big way that it contributes to genetic variation. That when those chromosomes line up randomly, it means you're going to get a new combination of chromosomes that were contributed by mom and dad. So let's take a look at that independent assortment. If let's say your homologous chromosomes lined up this way, so you had kind of all the blue ones on this side and the green ones on this side. When the homologous chromosomes separated in meiosis 1, and then the chromatids separated in meiosis 2, your ending result would be getting one of each of these blue ones in each cell, right? But let's just see, what if they flip-flopped? Okay, what if these two right here switched places and lined up just the opposite way? Well, now when these separate and then the homologous sister chromatids get pulled apart as well, look at this new combination you got. Okay? So it just depends on how those homologous chromosomes line up in the middle. It's not like all of your chromosomes that have the dominant genes go to the left side and the ones with the recessive genes go to the right side. It doesn't work that way. They line up randomly, so you can get, if you look at these possible gametes here, if these were the sperm cells or the egg cells, every single one of them has a different combination, don't they? That's where you get genetic variation. From there, your homologous chromosomes get pulled apart in anaphase 1. So again, remember, meiosis 1 is all about separating the homologous pairs. So they move to the opposite sides. You notice your sister chromatids are still attached by the centromere here, okay? So it's still a replicated chromosome. And in telophase 1, you notice that the cleavage furrow is pinching in right there. And your nuclear membrane's starting to come back. Your spindle starts to break down. Cell membrane begins to pinch in that cleavage furrow. And then we have cytokinesis 1. And again, that's going to divide that cell into 2 cells. Now, there may be a short phase in between meiosis 1 and meiosis 2 where the chromosomes temporarily kind of unwind a little bit, more proteins are made, but the DNA is not replicated, okay? So it's not a true interphase like our first interphase where DNA has that S phase, where the interphase has that S phase and the DNA is replicated. That does not happen here. So this is kind of a mini almost interphase in between meiosis 1 and 2 where, you know, we still maybe need to produce some more proteins and things, but we're not replicating the DNA. Meiosis 2 divides the sister chromatids, okay? So meiosis 2 will look very much like mitosis in terms of we're separating a replicated chromosome into its individual chromatids. Again, the DNA is not replicated in between meiosis 1 and meiosis 2. So in prophase 2, the chromosomes are again condensed and visible. The spindle forms, the nuclear membrane and nucleolus break down. So just like we do in mitosis. In metaphase 2, the chromosomes line up in the middle of the cell. Notice there's no pairs to line up like we had in metaphase 1 because we've already separated the homologous chromosomes. So they line up in the middle just like they did kind of in mitosis. Anaphase 2, the sister chromatids are pulled to the opposite sides. You see that happening here, okay? And then in telophase 2, the cleavage furrow forms. It starts to pinch in. So this is anaphase here, and this is telophase right here. Nuclear membrane starts to reform. Chromosomes start to uncoil. The spindle fibers disappear. And then we have cytokinesis. So that will actually divide the cell into 4 daughter cells that are genetically different, okay? And they are haploid cells that are genetically different, all right? So here we have meiosis 1. Again, prior to that, interphase happened and the DNA was copied. So we have 2 of every kind of chromosome in prophase 1. The homologous chromosomes pair up and crossing over can occur. Metaphase 1, the homologous chromosomes line up in the middle, and they do so randomly so we can have independent assortment. Anaphase 1, the homologous pairs get pulled to the opposite sides. Telophase 1, nuclear membrane forms around them, those pulled apart chromosomes, and the cell membrane starts to pinch in. Meiosis 2, it's at this point, these are going to be haploid cells. Okay, it's the reduction division. We went from diploid to haploid. From here, those in prophase 2, nuclear membrane will break down, chromosomes will thicken. Metaphase 2, the replicated chromosomes will line up in the middle of a cell. Anaphase 2, sister chromatids get pulled apart. And telophase 2, the cell membrane starts to pinch in, nuclear membrane will start to reform, and then we have cytokinesis, the second cytokinesis, and we're going to end up with 4 non-identical haploid daughter cells. So they are genetically unique, they are haploid, and these will be your gametes. Remember, meiosis forms gametes, egg and sperm. Okay? We give that term a name. It's called gametogenesis. That's the formation of sex cells or the formation of gametes. It's a little different between males and females. With males, if you have one initial germ, we call it a germ cell. That's what the diploid cell is. That's the somatic cell that's going to undergo meiosis. it will undergo 2 divisions. Here's one. Here's the first meiosis 1. And here's meiosis 2. And it's going to form 4 sperm cells that are haploid. Okay? The egg, however, has unequal cell division. So here's meiosis 1, and notice when it divided, one of these cells got most of the cytoplasm, and that would include most of the organelles and everything like that. And this one got very little. Okay? When this divides again, even if this divides, it would divide into 2 really small polar bodies. But what you end up with is you have one egg, but these 3 polar bodies, they all die because they don't have enough nutrients, because all the nutrients went to the egg cell. So for males, each diploid cell at the beginning of meiosis will produce 4 haploid sperm cells, and for females, each diploid cell at the beginning of meiosis will produce 1 haploid egg cell. So it's a little bit different for males and females. Males can also keep producing sperm throughout their lifetime and until they're much older, whereas females are born with as many egg cells as they're going to have, because those egg cells are formed actually while they are still developing inside their mom before they're born. So again, let's compare and contrast mitosis and meiosis. They are both forms of cellular division. It's just that mitosis produces genetically identical cells called somatic cells or body cells. Meiosis produces genetically unique cells, your gametes. Mitosis results in diploid somatic cells. Meiosis results in haploid gametes. Again, mitosis can take place throughout the organism's lifetime, where meiosis only occurs at certain times in an organism's life cycle. Mitosis is involved in asexual reproduction, whereas meiosis is involved in sexual reproduction with the formation of those gametes. So again, meiosis, 2 cell divisions. Mitosis only has 1 cell division. In mitosis, the homologous chromosomes never pair up. And again, meiosis results in haploid gametes that are genetically different. Mitosis results in diploid cells, somatic cells that are genetically the same. So you should really understand and be able to compare and contrast mitosis and meiosis. And the other big important thing is, remember that meiosis provides genetic variation. What are those 2 processes that occur during meiosis that provide that genetic variation, and when do these processes occur? Well, the first one is crossing over. And that's when chromosome tips of homologous chromosomes exchange genes, providing new combinations. Thus, new genetic variation. And it occurs in prophase 1. The second one was independent assortment. That's when the homologous chromosomes line up randomly in middle of cell and are pulled apart independently. Of each other. And that provides new combinations. And that occurs in metaphase 1. metaphase 1, prophase 1, independent assortment. So those are your two main types of processes that provide genetic variation. That's why in the end, our gametes are all genetically unique because they've had some crossing over events and they've had independent assortment occur.